Hi, this is a video for the TA guide for electrophoresis for Chem 202 Affinity Lab. First off, there will be directions here for the gel if you need it. Otherwise, we'll be walking you through it in this video. So students will come in with 4 already grabbed samples of protein. So they'll have their unknown, so let's say 74, and then they'll also have their FT, and then elution A and B, and then they should come in with all of these 4 proteins with 25 microliters pipetted into bullet tubes, and then in here they will micropipette another 25 microliters of the sample buffer with BME, just glue. And then from here, the TA's job is just to get it set up for the gel. All right, in room 213 in the refrigerator, on the bottom is gonna be, on the right side, your gels. Just grab one and go. I am now just getting the gel ready to put in. It's very fragile, so be careful. And then make sure to take this green tape off. Alright, now that we grabbed our gel from the fridge, we just opened it, and we're going to be taking this chamber out, if I can get it. And then what you're going to do, right now we are just running 1, but if you had 2, you're just going to put it on one side, and then the other one on this side here. Just like this. And then just put these sides up. I do like to have the numbers facing out just so you can see what you're pipetting in. And then I also, just a reminder to take this green tape off that bottom there, and then you're good to put it in. And then we will be filling it with SDS buffer, and you want to make sure to fill it in to the 2 gel line just because we have 1 chamber, but if you're filling it to the 4 gel, you need another chamber with 2 other gels inside. And then with this, you're going to fill it up from in here first until it overflows to that 2 gel line. Oh, quickly before I do this, we are just going to take this green part out as well. Looks like I'm going to need to grab another bottle. And there is a little bubbly, but I'm going to peek around a little bit more, otherwise it looks about good. Alright, and now the student should already have set up their proteins with the blue dye in it, and then you'll just have them sign up for the order they're gonna go in. So you as a TA need to make sure you're filling out all this top information, and then you are gonna have, you could either have a ladder and then 3 students, just because that's all that fits on here, or like a student, a ladder, etc. Just at least you fit a ladder in there for something the students are able to compare their data to. And then the students again should have their unknown number, FT, elution A, and then elution B, and then with the 3 students, and make sure they fill that out. Alright, now you're going to have your students micropipette everything into here. So you're going to want to grab the micropipette with the longer, thinner tip, just so your microliters will just kind of get up to here when you're putting it in, and it'll fit in the gel. And then for your ladder or your standardized comparison one, you're going to want to grab it, I believe, from the freezer. It'll say on this sheet here. You're going to want to micropipette 12 microliters of this, and then your students will micropipette 14 microliters of their own in each slot. So to do this, you are going to pull this up so it locks, and then when you twist it, it is at 12. So that's good. So I'm just going to push it back down. And then the proper micropipetting technique, when you put this in and push down, you don't want to push down all the way because you're going to soak up or grab up more than the recommended, or 12 microliters in this case. So what you want to do is just push down slightly till you hit kind of a stop, and then pull that liquid up. And then when you're in here, push it all the way down just to get all those air bubbles out. And then make sure to pull it back up out of the gel before you let go. Otherwise, you'll soak up all the gel in your micropipette and that whole gel is ruined. So now we'll actually micropipette. So this is just my standardized ladder. So I'm just going to push it down slightly, put it in, slowly pull up. Make sure there's no air bubbles. Okay. And then from here, slot 1 will be that right here. Right now we have our 12 microliters of the standardized ladder, and we're just going to put it in slot 1 and make sure you're in that gel here. And I'm pushing down, and then I am not letting go until I lift up. Alright, and now we're going to add the student's protein, or the students will themselves, and then you're going to make sure you're putting it in the order that they wrote it here, right? So this unknown 74 will go in slot 1, FT will go in slot 2, etc. In this case, we did start with slot 1 with our ladder, but we could just bump it down for the video. And then also make sure you're grabbing a new micropipette tip every time you dip it into a new substance. In which we did. And then for this one, you also have to make sure that you switched it to 14 microliters for the students' proteins. Alright, and now we are going to micropipette it slowly. Alright, there we go. And this would follow along with that slot, and you're just going to put it in. And then I'm making sure I'm pulling out before I lift up here, and then just dispose of that micropipette and grab a new one. We don't need to because we aren't, and I look, FT is next, so I'm making sure I'm following this FT label. Again, micropipette. No air bubbles. Alrighty, and then putting it in the correct slot. Slowly pushing all the way down. Come up, let go. Dispose of micropipette. Put on a new one, right? And next one is elution A, following the paper. Same thing, not pushing all the way down, but I am micropipetting it up. Closing it, next slot. Alright, lifting all the way up outside of the gel, disposing, new tip, elution B, following the paper. Alright, now I am going in, correct slot. Alright, and lift up, dispose, biohazard. And then, yes, that should be good. Alright, now that we have all of our gel set up, all the students' samples, proteins are in their slots, we want to make sure that we lined up with red to red and then black to black. And then we will be putting this on top, black, red, etc. And then just hopefully sliding it on. Alright, and then from here, you're just going to turn it on. So just on, make sure it's on volts, and then you're going to put it up to 140. Maybe you can speed this part along. Oh, oops. Pay attention, I guess. Perfect. And then you're just going to click run, which I won't do, but then you're going to check it every 10 minutes to make sure it's running properly. If you do not see bubbles coming up, it's doing it wrong. Try to check, see what's going on, and then about 30 to 40 minutes it should be done running. Alright, now that I checked every 10 minutes, it ran perfectly. It is about 35 minutes. I'm just going to press stop, and then I can turn it off, and then I can take this off. And then we just take the gel out and head over there. We grabbed our gel out, and now we are going to take the gel out. Kind of just cracking it open on each side over the sink. And then make sure to be very careful carrying this gel out. It is very fragile. Alright, as I'm struggling. Trying to see which side it's sticking on. Okay, gel is on this side. So now I'm just going to bring it over. Lightly pull it. Perfect. Set it in there. And then make sure to grab your aqua stain. Make sure it's not used, but for this purpose we are using used. And then just pour some in. A little bit more than that, making sure it's all submerged. That's a good amount. And then we're just going to put this lid on. Alright, now we have our gel in the solution. We're going to place it on here. And then you're just going to turn it up. Not too fast, but fast enough. And then the next day, you will take it out, take a picture, send it to the prof of that class, and then also send it to all the emails of the students so that they can get their data and complete their lab report. And that is how you do Affinity.